Hey guys, welcome to another video of Usman, and in this video we are going to talk about reduction in the chromosomal number by the meiosis process. How the actually meiosis process do the reduction in the chromosomal number to the half. But before we go through in these details, firstly we talk about the meiosis process, the overall scenario of the meiosis in just 1 minute. This is one cell that is producing the two cells. One cell producing the two cells, and one of these two cells that are produced also undergo the meiotic process and forming another two cells. Here one cell producing two cells. So it means that in an overview, in a big picture, one cell actually forming the four cells, and each of these formed four cells are the haploid. They are now the diploid. While the parent cell that is forming these cells, parent cell, is the diploid. It means that there is a reduction in the chromosome number to half. Here is the reduction. 46 number of the chromosomes are reduced to half. While the meiosis is divided into the two subdivisions, meiosis 1 and the meiosis 2. Meiosis 1 forming the two cells and meiosis 2 also forming the two cells. In a both scenario, one forming the four cells, and the chromosome number is reduced to half. This is what meant by the reduction in the chromosome number by the meiosis. Here we go about the meiosis in the females and meiosis in the males. Meiosis in the females, these oogonia are present, and they are the germ cells actually present in the female. Here is the oogonia. Oogonia undergo the mitotic process and forming the germ cells, and there are the million of the germ cells that is formed by the mitotic process of the oogonia. When these germ cells become matured, they are present as the primary oocytes. The primary oocytes are now able to enter into the next phase, that is called the first meiotic division. The primary oocytes enter the first meiotic division, but before the primary oocytes are able to pass through and completed the first meiotic division, there is a block present in them that stops the primary oocytes to go through into the complete stages of the first meiotic division. Here we see, here is the meiosis. The meiosis consists of the prophase, metaphase, anaphase, and telophase. There are the four stages that is consisting of the meiosis. While the prophase also subdivided into the different stages, but here the main stage is the diploid. Diploid. This is the stage where the block is present. The primary oocytes reaches at the prophase at the diploid stage, but there is a block present that stops the primary oocytes to go through, to pass through all of the remaining stages, that is metaphase, anaphase, and the telophase. So the primary oocyte stops here. They do not further develop under the meiotic process. Instead of this, however, this block is actually very important for them, because this is the place where all of the materials that are required in the later stages in the developing embryo. In the developing embryo, different things are required, and all of these things that are necessary, such as the yolk sac, and there is a genes that forming the different kind of the RNAs in the yolk sac that is required in the embryo, in the development of the embryo, are found here. This is the first block. This is the first block, and there are the two blocks that is present in them. Here, this is the second block. So due to this, only a limited kinds of the primary oocytes, limited number of the primary oocytes are able to complete the first meiotic division. Only 5 to 30 primary oocytes are able to complete the first meiotic division and forming the secondary oocytes. And this block actually forming the all of the materials that is required for the during the embryo development. So here this candiocyte is found, and these candiocytes are now entering into the second meiotic division. Second meiotic division. Second meiotic division. And this second meiotic division, before they complete the second meiotic division, there is a block present at the metaphase. Here, block is present at the metaphase. The metaphase stop these candiocytes to complete the further steps of the meiosis and halt the process, and the stimulus for release from these blocks is actually the fertilization. When the sperms comes in contact with the scandiocytes, they fertilize the scandiocytes. then this block is released and the scaniocytes completed the second meiotic division and form the fertilized ovum. But with the scaniocytes, another thing is formed, that is called the first polar body. First polar body. This is actually formed, the first polar body. And when the scaniocytes hit the sperms fertilize the scaniocytes and forming the fertilized ovum. Also another thing is formed, that is called the second polar body. Second polar body. This is what happening in the meiosis process. And only a limited number of the primary sites are able to pass into the scaniocytes, 5 to 30, and the scaniocytes are fertilized by the sperms and complete the second meiotic division and form the fertilized ovum with the second polar body. This is what happening in meiosis process in the females. If we talk about the meiosis in the males, the meiosis actually in the males consisting of the spermatogonia, these are called the germ cells. These are called the germ cells in the males. In the previous video we have talked about, they undergo the mitotic process, mitosis. They undergo the mitotic process and forming the different number of the primary spermatocytes. These primary spermatocytes are actually called the matured form of the spermatogonia. When the spermatogonia undergo the maturation, they are present as the primary spermatocytes. These primary spermatocytes now able to enter into the first meiotic division of the meiosis in the males. The first meiotic division producing the two secondary spermatocytes. These secondary the primary sites takes a time of the several weeks. They take several weeks to pass through and to complete the first meiotic division, but there is no block present in them. They take the several weeks and complete the first meiotic division and form the secondary oocytes. These secondary oocytes also passes through a second phase, that is called the second meiotic division. And the second meiotic division, under the second meiotic division, they completed the second meiotic division and form the spermatids. Here are the 4 spermatids are formed by this process. This is what happening in the spermatogonia in the males. While the scaniocytes only take the 24 hours to complete the second meiotic division. The primary spermatocytes are forming the 4 spermatids, and here the oogonia forming the fertilized ovum. These spermatids undergo another process that we are not talking here, that is the spermatozoa. The spermatozoa, after the maturation of these spermatids into the spermatozoa fertilize the scaniocytes and forming the fertilized ovum. This is the whole scenario of the reduction in the chromosome number by the meiosis process. So in the oogonia, here the scaniocytes are formed, and all of these scaniocytes are the haploid here, while the spermatogonia forming the spermatids, and all of them are the haploid here, while they are the diploid. Spermatogonia diploid, oogonia diploid. So they are forming the haploid cells. They are forming the haploid daughter cells. So this is meant by the reduction in the chromosome number by the meiosis. Hope you understand this. If you have any question regarding this topic, you can just go into the comments, and please subscribe and share this channel. Thank you.